phỏng vấn
So as we get the questions queued up, I'm going to start
3 nhà
because we have some students in the
học
audience
giải
with a general
tưởng
question
và
for
phân biệt
all of
trong
you. So
lĩnh vực hóa sinh.
we don't all know what we want to be when we grow up. Some of us have an idea that we want to be a doctor or an astronaut right away.
Quang bạn bắt đầu.
But how was it in your education and training? What moment was it that when you realized that you wanted to be a scientist and follow the career path that you've discovered? I'll give you each a moment to answer. Maybe start with you, Nick.
Yeah, good question. So for me, I didn't know what I wanted to do when I was growing up, unlike my sister at the time, who definitely knew what she wanted to do. I think you have to find something that you enjoy, you're invested in, and that you're passionate about. And I'm fortunate that I found that with science later on. But I think I also only really found that by trying other things that I wasn't passionate about. So having opportunities to do other things that maybe I thought I would enjoy and actually realised I didn't enjoy. And that really brought me back to science, where I always had the interest for, and then kind of realised, well, okay, well maybe this could be a career for me. And so there's an element of chance there and timing as well. So I think it's different for everyone. But if you find something you're really passionate about, then I'm really fortunate now that I go to work and it definitely doesn't feel like going to work. I get to study things that still fascinate me now. So it's wonderful.
Yeah, there's this effortlessness to doing work that you would want to do anyway because you enjoy it so much, right?
Absolutely.
Yeah. How about you, Pontus? Did you always know that you wanted to study ancient DNA?
No,
câu hỏi dành cho giải đồng.
I think I was always attracted by science, attracted to science early on in school, I think. But I don't think it seemed very feasible for me to become a scientist. That seemed very far off. But I guess it grows. It seems more and more realistic as the more you continue on, the more you sort of get into it, you pick subjects slowly. And so even though in the beginning it might seem like a big gap, it's more and more realistic if you follow what you're interested in. Or, I would almost say sometimes it's like you get very obsessed with things. It's an effort in that as well.
That makes sense. And what about you, Callie?
Yeah, so I grew up in Europe. So for me, it was never a career I considered. But I don't ask my mother, who was a high school maths teacher. So growing up, I was always interested in maths and science. And then in school, I had a chance to go to New Zealand and finish school. And a chemistry teacher pretty much used his extra time to mentor me, and that's where I realized that could be a good
Người Việt Nam đang ngồi đây.
Yeah, mentorship is so critically important. It's something I hear time and time again. So, I wanted everyone to know that you don't just sort of end up like this, that it's actually, quite a journey from primary school to where you've all been today. And related to your science, do we have any questions from the audience members? Would anyone like to ask a question? Yes, I see a hand back there. Yes.
Shall I? This is for Dr. Nguyen. My question is whether or perhaps how much it would make a difference if you had an even more powerful microscope in your work.
Yeah.
I'm asking because I'm invested in a project which is using quantum physics for the very first time to do photosensing, and it will be many more times powerful than, at every rate, including things like the Hubble Space Telescope, many more times powerful than anything that is currently in existence, but including if you go down to the smallest level.
It's a good question. It would always be better to get better instruments, I would say, because what we've done recently, it couldn't be done even 10 years back, just to see we are working really at the cutting edge of the technology. So to do this cutting edge research, you need cutting edge technology that evolves very fast for what we do. So every year there's always new updates, and we always try to get that and push a bit. The more details you can see, the more you can learn. and the more information you get from it, the better you get at making predictions.
I would just add, I don't know how far away this technology is. It's called extreme quantum imaging, and they're currently trying to build the first serious prototype, let's say. So it could be some time away. I don't think you'll see it in next year's update, let's put it that way.
But I keep my eyes out.
And thank you for your talk. Thank everybody for their talk, actually, all very interesting.
Yes, thank you so much for very interesting talks. It sounds also easy when you present it in a short number of minutes, and just the amount of work that has gone to get to the place where you are must be very heavy, especially so. Professor Nguyen, just thinking about you working on a molecule, you said if it's highly expressed, there's 50 of them in a cell, and then you're managing to isolate that along with all the stuff that comes attached to it. I mean, how long did this method development take you and your career, and what else can you do with it? I mean, I know you're invested in telomerase, but are there other really interesting proteins that are similarly rare and that you could now go and investigate, or your colleagues?
Yeah, that's a good question. So it's kind of a feature of my work, really. So I talked about telomerase, but I got into telomerase about 10 years ago, about almost 10 years ago when I started working on it. And there we could make progress in about 2 years for this quite long-term challenge, but I didn't go there from scratch. So in my PhD training, I worked with a machinery that has exactly similar problems. So it is called the spliceosome, that edit our RNA to make it into a mature message that the cell can read into a functional protein. So, and there I spent about 5.5 years going at this, and there were really, I didn't even have the work done by the end of my PhD, so I published it after. But again, all of the skills I learned through this journey that made the work for telomerase a lot easier, because then you've done all this training and you really had to think out of the box. You need to grow, for example, in my PhD I regularly grow 80 liters of yeast just to get really like a tiny amount of sample, and the same thing applies for telomerase. My students now are growing lots of cells to get just a few molecules, but that is sufficient. So it's a long journey, but a cumulative training that get us there in the end. Yeah. And regarding the later questions, what we would do with that. So I mentioned briefly at the end. So we're now thinking of finding ways to modulate telomerase. Besides a new avenue in my lab, which I haven't talked about, is looking at other repetitive DNA sequences. What is quite interesting is about 60% of our genome consists of sequences we usually think of junk. Because actually now, as we found out, they're not actually junk. They are remnants of our ancient history, like what Hans is talking about. So now we're looking at other repeat elements in the genome that make a vast majority of the genome. They are not genes, but now they're doing other functions in the cell we don't know. So that's very new. We only published the first paper last year, so hopefully in the next few years we have more work done on that.
Thank you so much for the talks. It was very interesting, and I have another question for Nguyen about the methodology. So, why cryo-EM specifically for the structural imaging of telomerase? Why wasn't NMR or X-ray or something else used? Because telomerase is coupled to a bunch of nucleotides, and wouldn't X-ray be more like informative? Thank you.
Yeah, the cryo technology has a few advantages for this kind of problem. So I trained in both NMR and X-ray. I couldn't tackle them with that, and that's why I moved to EM. So I've seen the whole range. So the big advantage is EM use a very small amount of sample, so that's one big advantage. Whereas you need like an order of magnitude more sample to do NMR and X-ray crystallography. Whereas in telomerase we got so little that we just can't use those methods in the first place. Second is that telomerase is very large and flexible. And the large and flexibility is difficult for X-ray crystallography because you need to grow crystal that has orders. On the other hand, NMR also can handle flexibility and conformation dynamics, but it cannot handle such a large molecule. So that's why EM happened to be the best. And technology in EM literally just took off 10 years ago. And I was happened to be right at the center of this development. You know, my neighboring labs are doing development, so we just happened to be at the right time, at the right place, and having a good sample to do this kind of, use this kind of technology.
Thank you. I have a question for you, Panos. As I listened to your talk, I had a few hints of this, but you know, you collaborate with historians and archaeologists and people across fields. Has your work actually altered our fundamental understanding of human history in any ways that you could share with us? Were there any things that you discovered that changed our knowledge of history?
Yeah, definitely, and there's so many instances of that also by other ancient DNA groups in the world. It's just, you know, for example, in history there will be historical sources, people that wrote things in the past. They have remained the same for, quite some time. Several decades, the same ones. People have been sort of delving into, like really trying to understand. But with ancient DNA, we are suddenly getting this entirely new written, if you will, record. So I talked about Scandinavia, for example, we can see just before the Viking Age, clearly a major influx of people. And this is before the historical period in Scandinavia itself. So it's something that we now have, and we try to kind of look at the historical and archaeological material culture records from the light of this and see, can we reinterpret the things that people have thought before of other changes that happened in this time period. So that's definitely an example.
And then from your study of ancient pathogens and ancient pandemics, what lessons can we take from history to apply to today and to the prediction of future pathogens and pathogen outbreaks, and how, what can we learn about how best to respond to them?
Yeah, very interesting question. Of course, we still need to get more data to understand those things, but what ancient DNA has revealed is many more outbreaks in the past than we had any idea of. For example, plague, Yersinia pestis, was present in the Stone Age, 5,000 years ago. Something we would have no chance of knowing if it wasn't for ancient DNA. But I think there's actually a very interesting thing, because our species, our genus Homo, is 2 or 3 million years old. But for much of that time period, its pathogen load or infection exposure was probably the same to other wild animals today, more or less. But 5 or 10,000 years ago, people started to produce their own food with agriculture, have domestic animals, live in denser settlements that created this entirely new situation of transmissible disease between people that I think wasn't previously in those previous 2 million years. So for a few thousand years there was this very different situation, and now, at least mostly in developed countries, the infection load is much reduced, particularly from bacteria. So there was this very big evolutionary jolt to our immune system, and the questions are if that is involved in many other diseases that we don't think of as having to do with infection today.
Oh, that's very interesting. So does anyone have any questions for Professor Casewell? I think I see one up here in the front.
Got a question from our virtual audience for
Great.
Professor Casewell. Are there any specific challenges about studying snake evolution compared to other venomous species?
Well, I mean, one of the advantages of working with snakes to study their venom is that these animals produce quite a lot of venom, and as you saw in the video, you can collect it relatively straightforwardly, some risk involved. But it is somewhat easier than trying to collect venom from a little marine cone snail or a small spider or a centipede. And so that has enabled a lot of snake venom research to kind of progress at pace and probably quicker than for other venomous animals. Simply you have more material to work with, and I suppose, analogous to some of the sample challenges that you face with your cryo-EM, having having more sample always makes life easier and enables you to do more things. So that's enabled venom research to progress. You can readily go and collect these venoms and work on them and have enough material so you can start to figure out what one protein does or another protein does and begin to compare them with one another.
And Nick, is snake venom, do snakes need venom for their survival and adaptation, or would another approach be to, I'm thinking about what Leslie Boschel has done with mosquitoes. So, she's developed a gene editing technique to basically remove mosquito-borne diseases. Would we want to consider something like that with snakes to make their venom less potent or maybe non-potent at all?
Yeah, I mean, it's an interesting concept, right? As you say, with vector control, people have tried to engineer transmissibility out of it so that infections can be halted. With snake bite's a little bit different in that there isn't this element of transmission. You have an event that happens, someone's bitten by a snake. But the biggest challenge is that these snakes require their venom to catch their prey. So they've evolved that venom system to enable them to feed, and without it they would obviously not survive. And you might say, well, maybe that's a solution, maybe we can actually just remove snakes from the environment too. But in many of these settings, they're really important animals. So they're controlling the rodent populations in these agricultural, rural areas of the tropics, where people are ultimately, their economic livelihoods relies on that. So there are ways I think we can mitigate the risk of snake bite, have prevention, reduce people's risk, but it'd be difficult, I think, to take a biological editing approach to it.
Yeah, that makes sense. Okay, so I'm getting the signal that we are out of time. But I know that you still have many questions. So the good news is that we are about to break for lunch. And I'm sure that our wonderful speakers would be more than happy to chat with you over the lunch break. Please join me in giving them another round of applause. So we'll be breaking for lunch. What time will we reconvene? We'll be back at 12:50, so there'll be an announcement at about 12:45 to make your way back into the auditorium for the next session. Okay. Enjoy.